In this portion, we'll talk about the anatomy and the physiology of the small intestine. So the small intestine is actually, when we talk about the structure of it, it is kind of in a way where it amplifies and maximizes the surface anatomy and the connection or the contact between the intestines, the wall of the intestine, and the chyme passing through it in order to have the most efficient absorption of nutrients. So the small intestine has what are known as circular folds, and within those circular folds, they have finger-like projections called villi, and inside of that villus, you'll find blood vessels, the artery and a vein, and you also find lacteals, which are lymphatics that help with in the absorption of fat and fat-soluble vitamins. The villi are covered with epithelial cells, columnar epithelium, that have microvilli, so that even further increases the contact between the lumen, the mucosal lining, and the chyme passing through it. The microvilli together are known as the brush border, and that's where you will find those final enzymes that are going to finalize digestion. So we'll take a look at the anatomy, and again, this is the inside of the small intestine with those circular folds, and here you could see a circular fold lined by those epithelium, those columnar epithelium. Here again is one of these folds. Inside the blood vessels, and in the middle you'll find that lacteal. A couple of things to kind of notice are those lymphatic nodules. So all of the nutrients that we're now absorbing, they might come in with germs, so the lymphatic nodules will try to trap them. You'll see on the lining, on these folds, we have again your simple columnar epithelium with microvilli. Again, these microvilli are known as the brush border, and this is where the final enzymes are found, like the sucrase, maltase, and lactase enzymes. You can also find a couple of other cells, for example, enteroendocrine cells, and those secrete all of the hormones that come out of the small intestine. And you could find scattered like goblet cells that make mucus. So any goblet cells that are found anywhere within the body would make mucus. Now for the pancreatic secretions, we already know that pancreas has both exocrine and an endocrine function. We already talked about its endocrine function. And we said that we'll discuss the exocrine function in the digestive system. So here we are. And the exocrine function would be the secretion of bicarbonate, mainly to neutralize the hydrochloric acid coming out of the stomach. And then there's also a number of digestive enzymes. The pancreas will pour them into the duodenum in order to help in the digestion of different things. So I'll show you actually in the next slide, which parts of the pancreas secrete what. I just want to kind of focus on this point in this slide where the enzymes secreted by the pancreas help in digestion of pretty much everything. So fat, polysaccharides, proteins, and nucleic acid, and it'll help break it down into its smaller components. So you can see here, this is just showing you kind of the relationship between the pancreatic duct and the common bile duct. The common bile duct carries bile from the gallbladder, and I'll kind of go over it since we have that on the slide right now. These are your two lobes of the liver, the right and the left lobe, and they both make bile. The bile will go through these right and left hepatic ducts, and both hepatic ducts connect together at that common hepatic duct. Bile will be stored in the gallbladder. It is stored and concentrated there. Okay, so bile is really made in the liver cells by the hepatocytes, but it is stored within the gallbladder. When it exits the gallbladder, it goes through that little tiny cystic duct. The cystic duct will then unite with the common hepatic duct to make a common bile duct. And that common bile duct opens at the common opening with the pancreatic duct and that gait is controlled by the sphincter of Oddi, and we kind of discussed the sphincter of Oddi when we were talking about CCK or cholecystokinin hormone. Okay, so that is just to show you kind of the relationship between that. And again, these would carry the digestive enzymes and the bicarbonate that are being made by the pancreatic cells. So now we can take a look at, focus a little bit more on the pancreas. And remember we said that we had the islets of Langerhans, those cells that make either the insulin and glucagon and so on. Okay, so that's not what we are going to focus on here. We're focusing on the bicarbonate and the digestive enzymes in this portion. Microscopically, the pancreas is made out of these acinar cells with ducts. Okay, so these, an acinar cell are cells that kind of look like a sphere, as you see here, or the gland looks like a sphere. And these secrete the digestive enzymes, while those duct cells secrete bicarbonate. They are stimulated by different things. So CCK, or cholecystokinin, will stimulate these acinar cells to produce more digestive enzymes, while secretin stimulates these duct cells to secrete bicarbonate. So if you go back and remember, from the beginning of the chapter when we were talking about secretin, we said that secretin is all about hydrochloric acid. It's all about the acid when it comes to this, where the stimulus to secrete secretin was the presence of hydrochloric acid in the duodenum. When it comes out, it will, on one hand, inhibit the stomach from making more acid, and it will also stimulate these duct cells to make bicarbonate in order to balance and neutralize that hydrochloric acid. Now, how do the pancreatic duct cells actually make bicarbonate? Well, again, this is your pancreatic duct cell. This is the lumen of the duct. And this is the basolateral membrane that is by the capillary. Within the pancreatic duct cell, carbon dioxide combines with water through that infamous enzyme known as carbonic anhydrase to make carbonic acid. Carbonic acid will then split into hydrogen and bicarbonate ion. Bicarbonate ion gets kicked out of the cell through an exchanger known as the chloride bicarbonate exchanger. So you see here that bicarbonate goes out and chloride goes in. Well, the cell doesn't really need that chloride, so it'll just exit out through what is known as the CFTR, or the cystic fibrosis transmembrane conductance regulator. And patients that have cystic fibrosis, they have a genetic mutation in the gene that is responsible for making that CFTR. And that is why one of the symptoms these patients have would be the inability to make bicarbonate in order to neutralize that hydrochloric acid. Well, what happens to that hydrogen ion that was made in that equation here? That hydrogen ion, again, gets kicked out of the cell through a sodium-hydrogen exchanger. The different pancreatic enzymes that are made, there's trypsinogen, chymotrypsinogen, and elastase. All of these are secreted in an inactive form, and then these enzymes will be activated in the small intestine through different enterokinases. So enterokinases, A's means that it's an enzyme, kinase means to activate, coming again from kinetic energy, and then entero means in the intestine. So all of these three enzymes that help in the digestion of protein are secreted in an inactive form, and they will be activated by these enterokinases found in the small intestine. You will notice that any enzyme that digests protein is released inactive because a lot of our body components, including the digestive system, are made out of proteins. So you don't really want these enzymes to kind of digest the system itself. You want to wait until there's food and in order to activate these enzymes and help in the digestion. So all of these digestive enzymes that act on proteins are going to break the peptide bonds to form smaller peptide fragments. Carboxypeptidase is another pancreatic enzyme that again helps in the digestion of protein, and it'll really split off that very terminal amino acid in that small peptide chain. Lipase, pancreatic lipase, digests triglycerides into a monoglyceride and 2 fatty acid chains. Pancreatic amylase helps in the digestion of polysaccharides into maltose. And then you have, last but not least, your ribonuclease and deoxyribonuclease that help in the digestion of nucleic acid into nucleotides. And here you can see here how the pancreas makes your inactive enzymes like trypsinogen, for example, that needs to be activated into trypsin, and it is activated again within the small intestine, not in the pancreas, in the small intestine through what are known as the membrane-bound entérokinases. And this ends this portion.